Good morning. Warm welcome from me, Cecilia van Heerden, for Memo discussion on paper 2 this morning. I just want to emphasize something on this memo to you, and that is when you look at the answers, many of these answers are straight from the exam guidelines 2021. For example, transcription, translation, DNA replication, out of Africa hypothesis, evidence of genetics for evolution, and it's actually straight out of the exam guidelines. It's stipulated like that in the exam guidelines and is also in the memo. So learners can really learn from the exam guidelines. On that, let's start then with question 1. Section A, question 1, multiple choice questions. And if you can look there on the memo, there is no alternative on this one. This is straightforward. I'm going to emphasize just to use something in queries that there was yesterday on some of the questions. So I just want to look at question 1.3. If you look at 1.3, we must remember now RNA is a single strand and DNA is a double strand, but the DNA have complementary bases. Remember now, adenine and guanine, adenine and thymine binds, and guanine and cytosine bind, so that must be the same. So if you look at this first table, W, X, Y and Z represent the nitrogen bases, which will be thymine, adenine, guanine and cytosine. So if you look at this one, molecule 1, molecule 2, 715, 715 is the same, as well as 523, 523. So this must be a DNA molecule because same amounts. It can be adenine and thymine or guanine and cytosine, or vice versa. And then molecule 4, also 103, and then these are the same. So these two are definitely DNA molecules. So 9A, that one is definitely RNA because it doesn't have molecules that are the same amount as with this one. Now you can look at this one and this one. So it's not a typing mistake, 7.8 and 8.7. That is there to confuse the learner or to put it on the next level grading of question. 9.5 and 9.5 is the same. That can be like that in an RNA molecule. It doesn't mean that they are the same or they bind. That is a coincidence. But those two are definitely your DNA molecules. So therefore, 1.1.3, we are going to highlight for you. 1.1.3 is then C, where molecule D is DNA and molecule 4 is DNA, and then the rest is RNA and RNA. So that is how we come to that answer. Level C answer, you have to imply your knowledge and then you have to calculate. So a bit difficult question on that one. And then also question on question 1.6. If you look at this question, you must look for the weak bonds, the weak bonds that binds nitrogen bases with one another. So if you look at this one, immediately I will eliminate A and D because they are strong bonds. You can see this one is a strong bond. There is the strong bond, so there it's a strong bond. But this one I have a weak bond, and there is the weak bond that we're looking for. So if we look at this one, B is the answer because the nitrogen bases bonds to the sugar. This one will not be the correct one because the nitrogen bases bonds to the phosphide. So the learner must know that nitrogen base bonds with the sugar, and it's weak bondings that is between them. So the answer is then B there in question 1.1.6. Right? If we look at question 1.1.7, learners must do a calculation. 4 different blood groups are possible when we cross A and B. So A will be heterozygous, heterozygous A, and B will also be heterozygous. Then we have 4 different blood groups: blood group AB, blood group B, blood group A, blood group O. So that is how we get to this answer. And in this case, the answer is C. A mother has a blood group AB, and the father has a blood group O. What is the percentage of them having a child with O blood group? So AB, there's AB, and there's the father. So in this case, they 0% chance of having a child with O blood group. All right. So just to emphasize on those multiple choice questions. So in this case, it stays the same as stated in the memo. And then there's no additionals at 1.2. Lamarckism, look at the spelling. Remember, the spelling must be correct, but if there's no other meaning in that, then you can take it as correct. Sometimes they struggle to spell Lamarckism, or they struggle to spell foramen magnum. But if it doesn't change the meaning of this word, then you can take it correct. And then look at chromatids. It must be chromatids. Chromosome will be there wrong. All right. And then question 1.3. B only, A only. If they write B, can be right. You can allocate the marks, or only A, you can allocate the marks. A and B, 2 marks, but A, B, 0 marks. Then we allocate 0 marks there. So just something to remember. 1.3.3, using in parenting testing is blood groups and DNA profiling. We use that, blood grouping and DNA profiling. Remember, blood grouping, we use it as an elimination. We know who is not the father, but we cannot definitely say who is the father, but we do use it in testing for paternity. Right. The next one, the next question on this one, 1.4, what is a karyotype? Or, the definition of the karyotype is the pairing and ordering of chromosomes according to their size and number. According to their size and number. So, in question 1.4, we have this one, where we have the question on this. So, give the definition. Give the number of autosomes. So, the learners must know autosomes are only up till here and there. And if I count it, normally it's 44, but in this case, there's an extra one, so it's 45. The type of chromosome, label A, it's a gonosome or a sex chromosome. The disorder is Down syndrome, and the process that causes Down syndrome is non-disjunction. The process is non-disjunction. So the answer is, in this case, stays definitely just like that. Question 1.5 on the roosters. They have to identify the genotype of the rooster. They have to identify the phenotype of the recessive characteristics and all the possible genotypes of the F1 generation. Also, they must look at that one. So, in the memo, as it states in the memo, we look for the characteristics. They are two, or they can write down the characteristics, feather color and comb shape, but remember now it's only one mark. So either 2 or 2. Then the rooster genotype, it's 2 marks or nothing. And then the black feathers and the unforked combs, each one is a mark. And the answer question was the phenotype of the recessive characteristics. Black feather and unforked combs. Be sensitive when the learner writes just black and unforked. So we need to know black feathers and we need to know unforked combs. So if a learner writes black, unforked, then you can decide at school, maybe you will take it correct, but emphasize then just for the learner that you will take it in this exam correct, but they must write actually what is the characteristic, the feather and the unforked combs. But let's say it's Christmas today, so we can allocate the marks there also. And then the last one is the possible genotypes. There it is. Those 3 we are going to identify. The learner will write another one. There's another one that can be like this one, but those 3 we need to mark. And in this case, you mark the first 3. Mark the first 3. Otherwise, they start guessing the answer. So that is on section A, question 1. So no alternative answers, but the mark, you can allocate marks there if you want to. Or it's correct, but you can just emphasize that it must be feathers at the end of the year. If we go to question 2, if you look at question 2, so identifying the structures, we have to identify the different structures here. C is the nucleus pore or nucleus membrane pore. In Afrikaans kan ons kernporie ook regmerk daar. Ons kan verseker kernporie regmerk. Dit hoef nie nucleusporie te wees, nucleus membraanporie nie, maar kernporie kan ons daar regmerk. D is tRNA. Molecule D is tRNA. Then name the molecule A. It's a double helix, so it's DNA. And molecule B is bRNA. Always important that learners must be able to identify this, and they can allocate the labels on this diagram. Tabulate 2 differences between the monomeres of the 2 molecules. Monomeres are also called the nucleotide of this question. And then describe transcription for 2 marks. So let us just look at the memo. So it's only the monomeres. So remember, in this case, we can only allocate marks for monomeres. And then a label or mark for the table. So in this case, long and short or double helix and single strand won't we are not going to allocate a mark there because we asked on the monomeres, and the monomeres are deoxyriboses and the nitrogen base. All right, and then 2.1.4, describe the role of molecule A. So molecule A is the double helix. So there it is. Molecule A is the double helix. So the role that the helix play, or the DNA plays in transcription is one string of DNA. One string. That is actually very important. Of the DNA molecule acts as a template on which complementary mRNA is formed. Or the learner could write one string of the DNA gives the code for protein synthesis. So, template on which mRNA is formed, or slash the code for protein synthesis. All right. Next question they ask in the question 2.2.1, state how many different mtRNAs is there. So there's one, and there's another one that is the same. So this one is the same, and this one is the same. Also this one and this one is the same. So at the end there's 1, 2, 3, 4, 5. Different types of tRNA. And then the normal questions: give the DNA triplet base sequence for the last codon of this mRNA. So this is the mRNA, and they want the DNA triplet of the last one. And the last one, DNA triplet, is then TAC. Remember also double marks in that case. You must write double marks in that one. And then the G stands for guanine. And then you have to explain DNA replication. In this case, it is very important to write it like this. The DNA double helix, not only double helix, not only the DNA unwinds, it unzips, two separate strands form, and then both the DNA strands serve as a template, build a complementary DNA. It's important to say a DNA because we are busy with DNA replication. We use 3 DNA nucleotides, not only nucleotides, but the DNA nucleotides, and it results in 2 identical DNA molecules. Each one consists of the original strand and 1 new one. So very important, the DNA double helix, 2 separate strands are formed, both DNA, complementary DNA, not just nucleotides, DNA nucleotides, and identical DNA molecules or DNA chromosomes must be there. Question 2.4.1. In question 2.4.1, we identify the parts. A is a chromatid, B the centromere, and C homologous chromosomes. Straightforward. Describe the process in meiosis, illustrated in the diagram. So if we look at that one, they have to indicate that it is crossing over because we describe the process. So the process is crossing over, and that is a compulsory mark. So at the end, it's very important, it's 1 plus 5 marks must be there. So what happens during crossing over? During prophase 1, homologous chromosomes, not chromosomes. It's not chromosomes. It must be homologous chromosomes. Overlaps at the chiasmata, or at the point called the chiasmata. Now this is very important. If they don't say the non-sister chromatids overlap, they can put non-sister chromatids there and say non-sister chromatids exchange genetic material. So why is sister chromatids so important, or adjacent homologous chromosomes. Why is that so important? Because remember, if they are lying like this together, and that one crosses, this leg and this leg is crossing over. And this leg of this chromosome and this leg of this chromosome are not sister chromosomes. This one and this one will be sister chromosomes. These two adjacent chromosomes are not sister chromosomes. So they must indicate which two chromatids are crossing over. They cannot just say the chromatodes exchange genetic information. If they say that the chromatids exchange genetic information, it means this one can exchange with this one, and that is not the case. So it's non-sister chromatids or adjacent chromatids that are exchanging genetic material. In die Afrikaans gaan ons sê homologe kromosome kom lê langs mekaar en nie sister chromatides oorkruis, of aangrensende chromatides van die homologe kromosoompaar ruil dan kromosome uit. Aangrensende chromatides. Nie net chromatides nie, want dit is nie wat hier gebeur tydens oorkruising nie. Dit is definitief non-sister chromatids, nie sister chromosome, of aangrensende sister chromosome. So be very sensitive. It must be homologous chromosomes, and who is overlapping or who is exchanging genetic material, non-sister chromatids or adjacent chromatids. That is very important. End of the year, they are very strict on this. You cannot just write chromosomes because it's homologous chromosomes, or that you can say when crossing over takes place, a chromosome from the mother and a chromosome from the father, but not just chromosomes. And then very important, which two are exchanging the genetic material on that one. 2.4.2 then. And then after crossing over takes place, we have to draw a diagram of the two chromosomes, and in this case, the homologous chromosomes after crossover. Not just homolog chromosomes, not just the heading chromosomes. So a mark for the heading. The color differences in the correct place. So there it is. And then if they draw two chromosomes, because they ask, draw a diagram of part C, and part C is both chromosomes. Can you see that? It's both chromosomes. And then that is where the mark is allocated, and there the exchange takes place. Nowhere else. And the tip change. This color of this one is at that tip, and this one on that side must be the same. Question 2.5. Question 2.5 was a question on genetic modify or genetic engineering, where they modify omega-3 fatty acids. Name the type of the fatty acid that is usually found in pigs. So that one, straight out of the paragraph, they can use it. And then they have to do a calculation. So the calculation in this case is 10 divided by 1,800. Give a mark. Multiply by 100. And then the answer, there is a variation. It's 055 or 056 also can be corrected. So because the answer is actually 5555, so we can round it off to 0.56. Right? And then give two reasons why some people may support the use of genetic modification in pigs that produce omega-3 fatty acids. also out of the extract, and healthier for humans to eat, mass production of healthy fat, improve intelligence. Remember now, we don't want a genetic or a general advantage of using genetic modification there. So that one of maybe it's cheaper, etc. In this case, it must come from the paragraph why we need this one. And then describe the steps in forming and introducing many copies of this desirable gene into pig embryos by using bacteria. So now we must take the general way of genetic engineering. When we use a bacteria of, you take the plasmid, you put in the desired gene, etc. Now we must take this gene and we must apply it to using bacteria. And therefore, in the answer, you must refer to the DNA replication as it is in omega-3 fatty acids. So the gene responsible for producing omega-3 fatty acid is located in the DNA of a tuna. Or they can also take the gene of omega-3 fatty acid out of the tuna, out of the mackerel, or out of the salmon. Then this gene, cut from the donor, is inserted in the plasmid of the bacterium. The plasmid of the bacterium. The bacterium replicates, and these genes are then inserted into the cell of the zygote or the embryo. So not a general state on how we use a bacteria cell in genetic engineering. Specifically on this example, you must apply the knowledge that you have. When we use a bacteria cell, we must apply it to the information in the extract. So that is on question 2.5. Just something, a comment on this one. You will see, especially in paper 2, you must have the basic knowledge, but with this we apply it to example. As the next question, we will have to apply speciation onto an example. So there they stated the story of the shrimps, snapping shrimps that can be found either side of the land bridge. However, when the males and the females from either side of the land bridge were brought together, they snapped aggressively at each other and would not mate. They are now considered to be different species. So this is speciation, and now we have to emphasize speciation or the knowledge of speciation onto the diagram as shown here. So the first thing, why is the inability to mate sufficient evidence to call the two groups different species? They cannot produce fertile offspring. So a species must be that when they interbreed, they must produce fertile offspring, and in the extract they stipulate that they cannot mate and produce fertile offspring. So now we come to speciation. So we must stipulate the original population of snapping shrimps, or naming this, the genus of the snapping shrimps, split into two populations by a geographical barrier, a land bridge, or and this one is compulsory, that you have identified the geographical barrier. So there's another mark, and they are very strict on this one. It must be the original population. the original population of snapping shrimps. Also, the original species of this same species of shrimps. But that one is very, very important to have in your answer. Okay, now there is no gene flow between the two populations of shrimps. So then I want to say something just here. The new way that I pick up in June, that I pick up when I'm marking this, is absolutely to go and apply speciation on the example. So when we say there is no gene flow between the two populations of shrimps, I want to ask or emphasize when you come back with the learners and you do corrections on this paper, maybe we must add, or not maybe, I think we add this now, because I pick up a trend that I want to mark it like this. So we are saying there is no gene flow between the two populations. Mention which two populations. The population of North America and the population of South America, because now we have divided it. Or if it was an island 1 and 2, then we indicate the two shrimps population cannot interbreed in island 1 and island 2 because of the geographical barrier. So just add there then, there were no gene flow between the two populations of shrimps between North America and South America. Now, the next one, since each population may be exposed to different environmental conditions, if the example explains different environmental conditions, maybe they say in the north the water is warmer and in the south cooler, then also add it in your question. Since they have exposed to different environmental conditions, mention the different environmental conditions. So I'm just emphasizing this for the end of the year. But status quo with this one, as it is stipulated in this example, we are going to write it like that. And then natural selection occurs independently. That word must be there on each of the two populations. And then I will add it in the end of the year at the North America and South America. Such individuals of the two populations become different from each other if they may be stipulated how they are different, maybe in color or maybe in size. Mention the differences also here for the end of the year. And even if the two populations were mixed again, they will not produce fertile offsprings, and this become different species. And now definitely here at the end, for the end of the year, you must say how different they become. They become different in species. We have the North America snapping shrimp and the South America snapping shrimp. So definitely name now the two different species that has formed there, please. So just to put this in context again, we have to say the original population of snapping shrimps. That must be there. You must indicate that they are split in how many of them are, and how many groups, in this case 2, and what was the geographical barrier. What was the geographical barrier? Then there were no gene flow between them, and the sequence must be like this, as stipulated in the exam guidelines. As stipulated. But as I said before, I think, or the trend that they are moving in, as I pick up in June, is more to come and put the example in your answer. So in this case, I will refer to the north and south. There is no gene flow between the two populations of shrimps in the north and in the south. So just add your example there. There's a lot of these examples in the past September question paper 2, 26, 2017, 2018, 2019, that you can go and practice that with your learners. Question 3. In question 3, also always in the exam paper 2, is differences and similarities between humans and African apes. And in this case we have the human and the gorilla, picture of that, and also a picture of the pelvis. Now the first question, state 3 characteristics of the upper limb that are the same in both species. So we're only going to look at the upper limb, and the similarities that you can use there is 3 rotating arms, long upper arms, finger 5 digits per limb, flat nails instead of claws, and opposable thumbs. I just want to say something here. Learners tend to write long arms. That is wrong. It must be long upper arms. Or they say they will only write 5 digits. Where? It must be 5 digits per limb. Flat nails instead of claws. Also, the other one that we can put in here is the elbows that allowed rotation of the forearms. Of by die Afrikaans kon ons ook sê, dit besit ellembooggewrigte wat die voorarm kan laat roteer. Elbow, it allows elbow rotation so that the forearms can rotate. So you can add that one also there. From the diagram, explain the significance of a visible change in the skeleton to prove bipedalism. So in this case, in A, the skeleton changed from a long and narrow to a broad and wide pelvis. And the significance of that is to support upper body weight. So that is the 3 points that we are allocating there. The pelvis changed from long and narrow and short and wide to support greater weight of the upper body. But long and narrow must be there. Short and wider must also be there. 3.1.3, 2 Australopithecus fossils that were found in Africa is the Taung child, Mrs. Place, Karabu, and Littlefoot. And we mark first 2. We mark first 2 only. Alright, and then explain how genetic evidence supports the idea that all living humans have common ancestors. So that is the out of Africa hypothesis, or genetic evidence of human evolution. And then we are going to refer to the mitochondrial DNA. Mitochondrial DNA. So you have to name mitochondrial DNA as the genetic evidence. Analyzed, have mutations on them. And these mutations sometimes act as markers. And these markers we can trace back to Mother Eve or back to the mother from which we descend. So mitochondrial DNA have mutations, and we can track these mutations because they act as markers. The word they act as markers. You can also allocate a mark for that. And these markers we can trace back to Mother Eve also. Right. Then the next one is explain this diagram, or give the name of this diagram. This is a phylogenetic tree. It's not a cladogram, because the cladogram must have characteristics with it, and then it's called a cladogram. In this case, it's a phylogenetic tree. State how many of each are the following. How many genera are there? There's Australopithecus, 1, and then there's Homo habilis, another one. So there's 2 genuses, and there's 7 species. 1, 2, 3, 4, 5, 6, 7 species in this question. Explain why Robustus and Bossii are more closely related than Bossii and Afarensis. So in this case, we must indicate that Robustus and Bossii share a more common ancestor. They share a more recent common ancestor, or other one that I also can mention is that the Afarensis ancestor lineage gives rise to 2 daughter lineage, which is Robustus and Bossii. But I think the more general answer that the learners will say is they have a common ancestor, but please emphasize it must be a more recent common ancestor. Which one of these are using tools, Homo habilis or Ha habilis? If they didn't write it like that or didn't underline it, we're going to allocate the marks so they can write. They can write Homo habilis just like that, and we're going to allocate the mark definitely there. Explain how the location and the age of Homo fossils are used as evidence of the out of Africa hypothesis. So the location and the age, you must refer, and only to Homo fossils. Not the whole out of Africa hypothesis, it's only the Homo fossils, and the Homo fossils explained in the exam guidelines is Homo habilis were only found in Africa. Homo habilis, so there is Homo habilis. So you refer to your Homo species. Genesis, the Homo habilis is only found in Africa. Then the oldest fossils of Homo erectus were found in Africa. The oldest fossils of Homo erectus is found in Africa. Now remember, the question is the location, Africa, and the age. The oldest fossils of Homo erectus were found in Africa. Then the oldest fossils of Homo sapiens were found in Africa. So in this case, when the learner wrote it in one sentence, he will write the oldest fossils of Homo erectus and Homo sapiens were found in Africa. Remember, then in that one sentence, it's 2 marks. While younger fossils were found in other parts of the world. So younger fossils are found in other parts of the world, implicating that because of the older ones that is found in Africa, that is why we have the out of Africa hypothesis. So this is actually stipulated in the exam guidelines just like that. So try and emphasize this to the learners and explain to them. In this case, they just want to refer to the genesis of Homo and how Homo fossils contribute to the out of Africa hypothesis. Not the whole sentence of out of Africa hypothesis. So the question may be now, what must you do if the learner starts with Ardipithecus is only found in Africa and then Australopithecus? All right, you read through it, and that is not applicable to the answer. You take cross it, and then you start marking here. So if the learner wrote the whole thing of out of Africa hypothesis, good for him, but we only allocated marks for Homo, the part on Homo, the species of Homo. But don't penalize them of writing the whole thing. So in this case, we don't mark the first 3 or whatever. We are going to read through the answer and allocate the marks. Again, on this one, as on mitochondrial DNA as a genetic, in the exam we found that learners cannot explain themselves and cannot give the explanation of the out of Africa hypothesis, so it's very important. And you can see the allocation of marks, 4 marks there. Question 3.3, the question on natural selection. They give the snail population show a variety of black shells and white shells. The characteristics of the cell color allow them to survive better in the environment, hence they were reproduced to pass on the favorable characteristic. The phenomena is called natural selection. So identify this phenomena first. Who identify this phenomena first? And this one was first identified by Charles Darwin. So what happens if the learners only write Darwin? There's another person, name is Erasmus Darwin, so then I'm going to 0 marks because it's Charles Darwin. It must be Charles Darwin. State the 4 characteristics for the snail population to survive. State the favorable characteristic was those with black shell have the favorable characteristics. And then the next one is describe 2 different types of mutation that may cause variation in a population. 2 types of mutation is first of all a gene mutation and a chromosome mutation, and then describe this mutation. Gene mutation is the change in the nucleotide sequence of a DNA molecule. Change in the nucleotide sequence or change in the nitrogen bases of a DNA molecule. And chromosome mutation, the result in the number of or the structure of the chromosome that is changing during meiosis. So that is the 4 marks we allocate there. Tabulate 3 differences between natural selection and artificial selection. There is the natural selection one. The environment is the selective force, the environment. And humans are the selective force with artificial selection. The selection is a response to suitability to the environment. So if the learner wrote here also, he can stipulate favorable characteristics. If an organism has the favorable characteristics, that one will survive, or the fittest will survive the environmental selective force. And in this case we say selection is a response to satisfy human needs. So in artificial selection, what human needs. And this one is which one survive in the environment. Of course, within one species, and this one can involve 1 or more species in artificial selection. So there will be learners that wrote it's a long process and it's a short process. According to marking at the end of the year, they don't allow that one because they say it can also take long in artificial selection to get the breeding that you want. So don't go with long and long period, short period. So let us leave that one out. The last question on question, on this question of natural selection or of paper 2 is the one on genetics, and then in this case we have the phylogenetic tree. There's the phylogenetic tree, and they ask, give the phenotype of individual 5. So there's 5. But we have to read through the paragraph. First of all, they say that this is a autosomal dominant gene. So the gene that causes this mutation must be dominant, and if you are unaffected, then you don't have the dominant allele, so therefore the female is capital letter D, small letter d. Phenotype, the genotype of individual, the phenotype is female without polydactyl or unaffected female. We must try to get away with this one. So explain what is in the table or at the key. So in the next question, genotype of individual 2, the individual 2 is heterozygous dominant, so D. Number of offsprings of parent 1 and 2, there are 6. There are 6 offsprings. If you look at this, 1, 2, 3, 4, 5, 6. That one and that one is not the offspring. Just to test if the learners know how to apply the knowledge on pedigree diagrams. And then the question is, explain what is meant by autosome dominant gene. So we are allocating a mark. If they can allocate that autosome chromosomes are from 1 to 2. And now the dominant gene, they have a dominant gene, that mutation for polydactyl, and will therefore be expressed in the phenotype only in a heterozygous or homozygous condition. So autosomal dominant allele means it's an allele that is found on the chromosomes 1 to 22. It is a dominant one that causes polydactyl, and in the phenotype it's only expressed in the heterozygous or the homozygous condition. So a lot of explanation there also. So that is on question paper 2. I think the marking guidelines is straightforward, maybe marking speciation and marking out of Africa hypothesis and marking this one. You have to interpretate, but the rest are really straightforward. As I indicated, those things that must be in the exam guidelines, be careful that the learner can write and it sounds right, but we need those key points that I emphasized to you in the paper. See to it that they know what to do there. And then the last one was on the crossing, using the letters D and small letter d. Now it's not on an X. It's not, you can, if the learner put it on X and Y, then the total crossing will be wrong. We allocate then only marks for P1 and F1, as well as meiosis and fertilization. P1, F1 gives a mark, and meiosis and fertilization gets a mark then, but then the other one is wrong. Just look at the compulsory mark. The compulsory mark is 1 to 1, because the question asks, give the ratio in the offsprings of individual 10 and 11. So that is a compulsory mark. They can also write it in this way, where they use the Punnett diagram and the compulsory mark. We must allocate a mark for the compulsory mark also. So if there's any questions, feel free to ask or send the SMS to your subject advisor. And if there's alternative answers that is coming up from learners, make sure you ask if you are uncertain on this one. But at any stage, I think the memo is really straightforward. And as I indicated, just please make sure we need those keywords, especially with DNA replication and crossing over, so that you have the right scientific answer in your question paper. From my side, thank you very much, and good luck with the marking, and stay safe, and thank you very much.